this video, we are going to explain species diversity analysis, alpha, beta, and taxonomy. Alpha diversity is the diversity of the sample within the sample. Beta diversity is the difference between samples or groups, including PCOA and CPCOA, species composition, relative abundance of different taxonomic level. Alpha diversity index is the method for evaluating alpha diversity, commonly used richness, observed OTUS, Ka1, ACE to estimate the number of sample species. This is the formula used to count singletons or doubletons. Shannon linear diversity calculates the Shannon entropy of each feature. Dominant index, Simpson 1 to dominance. The alpha diversity command calculate 14 kind of alpha diversity index calculation based on standardized OTU table. The SK biodiversity alpha package in Python 3 provide 34 alpha diversity index. You can use this command to go in detail about this. If we come back to our RStudio, and we can, because already in the normalization step, we generated alpha folder, and inside that we have already OTU tab rear text, begin text, OTU tab rear text, and OTU tab rear stat, which is going to use in the downstream analysis for plotting alpha diversity and calculating the diversity. Basically use users to calculate 14 alpha diversity indices. We will run this command here. And in order to calculate rear fraction richness, dilution curve, take the number of OTUs for sequencing ranging from 1% to 100%. We will use this command, and it's processed here. Now, in order to preview the result, we will run this command. You can see here the richness for each sample is given here, and handling of non-numeric values due to low sample sequencing, we use this command. Alpha text, alpha rear text. We can come here. We can simply check this. Berger, Parker, Bozas, Gibson, Ka1, dominance, equitability, Jost 1, Reed, richness, Robbins, Simpson, Shannon, Shannon 2, Shannon 10. And then this is alpha rear text, richness, and this is our samples. Come back to the RStudio. Now, filtering for high abundance bacteria, calculate the mean of each feature, and if there are groups, calculate the group mean. Group column name should be modified. The input file are the feature table, which can be OTU tab text, and the experimental design metadata text. The output is the feature table with group-wise mean. A single experiment may have multiple grouping method. The H in the command option display the script help, providing explanation for the parameters. We can run this command. And in this command, scale determine whether to perform scaling, and zoom represent to normalize the total SAM. All represent output mean for all samples. We will run this command, and the output is OTU tab mean text. We will see here this OTU tab mean text. The output include both the overall mean and the mean value for each group. Now, by filtering with an average abundance threshold of greater than 0.1%, which can be selected at 0.5% or 0.05%, you will obtain the OTU combination for each group. We will run this command. It will give us unique total group KO 182, OE 188, and W2 is 171. And you can use the alternative link here provided to draw the Venn diagram by using image GP. We will discuss this later. We can by using this link, we can create Venn, upstream view, and Sankey diagram. Now, come to the beta diversity. In beta diversity, the result generate multiple files and require a directory. First of all, we are going to create a folder inside the result with the name of beta. Click this command. Now you can see here it's beta. And then in order to build an evolutionary tree based on OTUs, we are going to generate some files which are compulsory to generate tree. Now it is going to give OTUS tree here. And now generate 5 distance matrices, including Recruiters, Elucidian, Jacquard, Manhattan, and Unifreight. We will run this code to generate these 5 kind of matrices. We can come over here. Now see, inside the beta we have multiple files. Now we can go to them. Break rotas sorted text. You can see here, this is break rotas. Column is also in the rows, and then you can see here the text, break rotas text. And now elucidation sorted text. You can see here all these files are generated to in result we will get the visualization, and then unifrac text. So you can go and you can see each of one by one. We will come back to the RStudio, and then species annotation and classification. User species annotation file result OTUS syntax, feature classification and confidence, the result after feature screening. It can be adjusted to the following standard format. For example, OTU corresponding species annotation in a two-column format. Remove confidence value from syntax. Retain species annotation. So we will get here taxonomy to text, and it will use OTUS syntax input as an input file. You can see here taxonomy to text. OTU corresponding species in an 8-column format. Generate species table where blank in the OTUs or ASV are filled with unassigned. We will run this command to get taxonomy text, and the input will be used OTUS text. You can see here. And then in order to count phylum, class, order, family, and genus using the rank parameter PCOFG, we will run this command. You can see here we are using OTUS syntax, and the result will be all these files, some text file. Now inside the taxonomy, it creates a folder inside the result with the name text, and inside the text we will get all the same text file. We can see here. You can see some C text, some F, some G, some O, and some P. List all the files by running this command. You can see here genus, unassigned, and this all our samples. Now reference-based quantitative feature table. Here we are going to perform alignment against the green genes, 97% OTUS database used in Pinterest bug-based functional prediction. Green gene database is the oldest, most accurate, and most widely supported database. Based on it, OTUS table, functional prediction, and morphological prediction can be performed. So first of all, we are going to create subfolder inside the result with the name of GG. There are several methods to perform this. Method 1 is using research, and method 2 is research alignment. We will go with method 1. And it takes several seconds to complete. Now 72.4% matched. Now it's still running, and we can see here the alignment rate at the end when it's at 10. As I already explained that using USET for alignment is faster, but encountering file size limit error. And if you use method 2, research alignment is more accurate but slower. However, it exhibits strong performance with parallelization using 24 to 96 threads. And in order to get statistics, we will run this code. Sample size minimum, low, medium, mean, high, maximum. Now, the next step is space cleanup and data submission. To delete the intermediate large files, we will run this code and calculate MD5 values for peer and data to use for data submission. Now we can see here MD5 sum text, and inside the GG